Okay, so this is going to be a video hopefully to explain all of those processes with all the different viral genetic makeups. So let's introduce some colors that I'm going to be using throughout this whole video, okay? So the first color I'm going to be using is this nice dark blue, and this is going to be DNA. And our nice dark blue is going to be the positive strand of DNA, okay? And then DNA, when it's double-stranded, has both the positive and the negative strand, and our negative DNA is going to be red. Okay, so let me get a black marker. So blue and red, this is DNA, okay? Those are the colors we will use for DNA. And this is double-stranded DNA, okay? So it's made of the positive and the negative. When we're going to be talking about RNA, we're going to use different colors. So RNA in this video, positive RNA is going to be green, and negative RNA is going to be pink. And I hope you can see these colors on the video. And so our pink and green is going to be strands of RNA. Okay. And then we have some enzymes. So what enzymes are we going to be talking about today? We're going to be using, I'm going to do a little triangle, and that is going to be DNA polymerase. And remember, a DNA polymerase takes a DNA strand and it makes another DNA strand. So if you want to go from DNA to DNA, you use DNA polymerase. We also have an enzyme. We're going to do a circle, a pink circle, and that is going to be RNA polymerase. And when we want to go from DNA to RNA, that is the enzyme we use. And this process specifically is taking DNA making mRNA, okay? That's the process that RNA polymerase does. These two enzymes are found in eukaryotic organisms. So if you are a eukaryote being infected by a virus, the virus is going to use your own enzymes. Now, we have two more enzymes that are specific to RNA viruses only, and they are, we're going to do an open square for Rna Replicase. And Rna Replicase takes RNA and it makes more RNA. Okay, it takes a strand of RNA and it makes it into another strand of RNA. So it's going from RNA to RNA, Rna Replicase. Eukaryotes don't have this enzyme. So if this is an enzyme that the virus needs to bring with it, okay, you're not going to find it in eukaryotes. And then the last enzyme that we're going to talk about, we're going to use a little star for this because it's a very strange enzyme. It's called reverse transcriptase. And it takes RNA and it turns it into DNA. This is our retroviruses, okay? Retroviruses need this. And it's not found in eukaryotes because in eukaryotes we never go from RNA to DNA. It's always the opposite. That's why we call it reverse transcriptase. It's going from RNA to DNA. So if you were an RNA virus, you need to bring these enzymes with you because your host is not going to have them. All right, so what are we going to learn about? We're going to learn about the following processes. We're going to learn about how we have a virus that is double-stranded DNA. How does it infect host cells? We're also going to be talking about single-stranded DNA viruses. We're going to talk about negative-stranded DNA viruses. So these are all our DNA viruses. We can have double-stranded DNA virus, we can have single-stranded positive DNA virus, and we can have single-stranded negative DNA virus. And then we're going to talk about the RNA. And so what kind of RNA are we going to talk about? We're going to talk about double-stranded RNA viruses. We're going to talk about single-stranded positive RNA, and we're going to talk about negative single-stranded RNA. So these are our RNA viruses. We can have double-stranded, we can have single-stranded positive, and we can have single-stranded negative. And then we can have retroviruses, which are technically a type of positive single-stranded RNA viruses. They're a type of them, but with a twist. So with a little twist. So these are going to be our retroviruses. And they are technically a positive-stranded single strand of RNA, but they are retroviruses. They don't act like our typical single-stranded positive RNA. They act differently. HIV is an example of a retrovirus. All right, so we're going to jump in. Let's do the DNA ones first, okay? So we need to keep track of what we start with, what we end with, and the enzyme players that are going to be playing in this game. So as long as you memorize the enzymes, what they do, you should be able to, if given a scenario, be able to map out what happens based on these big rules with the single and positive strands and all the different enzymes. Okay, so let's start with the DNA ones. Get a nice new sheet of paper out. All right, so let's set this up. Okay, so what I'm going to do first is I'm going to make a line on this paper here in pencil. And this line is going to represent the cell membrane of the host that we're invading. Okay. So this is just going to represent that membrane that the virus has to go through to get. So this is the outside world here, and then once it crosses over the membrane, this is the host cell now, okay? This is the host cell side. All right, so let's just start with one and work our way down. Okay, so we're going to be doing our DNA viruses. Now, where is that other piece of paper I just had with all of the player names on it? So one more concept before we start. This was in the lecture. Okay, so the virus is going to infect a host, and the host is going to have machinery called the ribosome. And it's formed of two units, a small unit and a large subunit, and this is a ribosome. We've learned about what they do. They make proteins. In this analogy, our ribosomes are the chefs making the recipes, and then the recipe that it needs is always mRNA. mRNA is also known as positive single-stranded RNA. So if you see this, it's the same thing. So mRNA is the same thing as positive single-stranded RNA. And if the ribosome encounters this, it can go through the ribosome, and then the ribosome is going to make the products. So the ribosome comes from the host, this comes from the virus, and it has to be in this language. It has to be in this language, otherwise the host ribosome cannot read this. So if it is in any of these languages, if the virus are any of these languages, it needs to be converted into this language first before the ribosome can read it. So that's always a goal that we're looking at, because the ribosome is going to make the capsid. It's going to make the capsid proteins. That's the outer shell of the virus. It's going to make that. It's going to make the spike proteins that go on the outside of the virus. If there are any enzymes that need to be packaged in with the virus, the ribosome is going to make those as well. So it makes all of those products, and then the products come together. And they're also going to grab the duplicated genetic material, which we haven't talked about yet, but that's the second process. And it's going to get packaged in here, and then this virus can go on and infect other host cells. So we need to make these pieces, and the way we do that is by taking the virus genetic code, making sure it's in this language that the ribosome can read, and then the ribosome will make all the parts. Getting to this is the complicated part. It involves the enzymes, and it involves thinking about the positive and negative strands when we're duplicating genetic material. So it has to be in this form, otherwise nothing will happen. Okay, so we got that clear. Let's then dive into what I was talking about. So we're going to do DNA first. Okay, let's get all the pens that I need ready so that I can just grab them without taking off the lids while I'm working. All right, we're ready. Okay, so let's start off with a double-stranded DNA virus, okay? So our double-stranded DNA virus, remember, double-stranded is made of two parts. So we've got the positive strands, and we have the negative strand, and they're twisted together, and this is our double-stranded DNA. So we've got double-stranded DNA. And this is the virus. This is the virus genome, okay? And it's going to infect the host cell. So what happens is the genetic material goes into the host cell. Now, what do we need for the ribosome to be able to read the genetic material? Can the ribosome read double-stranded DNA? No, it can't. It needs to have the single-stranded positive RNA. So how do we get from here to that? It's pretty simple. So what's going to happen is we need to make a positive strand. So the rule is that you have to have the negative strand as a template to produce a new positive strand. If you want a new negative strand, you have to use the positive strand as a template. And in a double-stranded DNA virus, we have both strands. We can pick and choose which one we want because we are given both. So we want to make more positive. So what we're going to do is we're going to take the negative strand and we're going to open this up and we're going to use the negative strand as our template, okay? We want to go from DNA, eventually we want to get the mRNA. So we're going from DNA to RNA. So what enzyme do we need if we're going from DNA to RNA? We need Rna polymerase. So Rna polymerase is going to come in and it's going to attach to the template strand, and we're using Rna polymerase. So we're going from DNA to RNA, Rna polymerase, and it's going to make a strand, a positive single strand of RNA. And that's the goal. That's what we need. So that was only one step, and we only used one enzyme to get there. Pretty okay. So now what do we do with this? Well, this can now go to the ribosome. It's ready to go because the ribosome can read this. That's the language it needs. We're aiming for green. And then what happens is it reads, and we get the capsid, we get capsids, and we get spikes. And we're done with that step. Simple enough. The second goal is to duplicate our original form of genetic material. So the virus started off as a double-stranded DNA. We need more double-stranded DNA to package, into the capsid. Okay? I think the computer turned off. So we need to get more of this double-stranded DNA to package into here. So we're starting off with double-stranded. There's our positive and our negative strand, and we need to copy both, and we are going from, we want double-stranded DNA, we want to end with double-stranded DNA, so we're going from DNA to DNA, so we need a different enzyme. So what happens is that this opens up, and both strands are exposed. And now our negative strand can make a new positive strand. And our positive strand can make a new negative strand. So we get a new positive and negative strand. Now what enzyme comes into play for that one? If we're going from DNA to DNA, it is the DNA polymerase, which was the little triangle enzyme. So we're going from DNA to DNA, so we need DNA polymerase. It attaches to each of the template strands and it makes a copy. And so now we're good, we're done. So we've made more of the genetic code, and then that genetic code can be packaged into the components that we made up here. And then new virus is free to go infect other cells at that point. So that's double-stranded DNA viruses. Now let's do positive single-stranded DNA viruses. Okay. So we're not going to start off with two strands. We're only going to start off with one strand. What strand are we starting off with? We're going to start off with that blue positive strand, and that's all we got. That's all the virus was packaged with, and that's all the virus is going to insert into the host cell is this single strand of DNA. All right, so it crosses over into the cell, and what do we need to make? We need to make, remember, we need to make single-stranded positive RNA. How do we go from single-stranded DNA to positive-stranded RNA? So if you remember the rule, if you have a positive strand, it can only make a negative strand. So this strand cannot directly make another positive strand, all right? It needs another step in between. So because positive strands only can make negative strands. So what we first need to do is we need to take this DNA, the single-stranded DNA, and make a negative strand. We need to make a negative single strand of DNA. So we're going from DNA to DNA. DNA to DNA, what enzyme do we use? We used DNA polymerase. It comes in, it sits on the template, and from that template, it then builds the negative strand of DNA. So now we have a negative strand of DNA. Negative strands will make a positive strand, so we're going to go from the negative DNA strand to the positive RNA. We're going from DNA to RNA, so what enzyme do we need? And when we go from DNA to RNA, we need Rna polymerase. Rna polymerase sits on the template and it makes the positive strand, single strand of RNA. And there we go. This is what we need. So we had one step in between getting there, just because of the rule that positive strands can only make negative and negative can only make positive, and we needed to go from a positive to a positive, so there needs to be a step in between. There needs to be a step in between because we can't just duplicate a positive and expect to get another positive out of it. So there's just one additional step here for that. Why did the first example not have to have that second step? Because we were given both strands and we could choose which one we needed. In this case, the virus only had a positive strand to give, and that's all it had to give. So then this can go into the ribosome and make the capsids and the spikes. It makes all the products, all the viral particles. So that's the first step. Now we need to make more of our original form to package into those capsids. Okay, so that's the second goal. So we take our single positive strand of DNA and we need to make more of this. Can we make a copy of a positive strand directly to another positive strand? No, because that's not how that works. Positives can only generate a negative strand. So there's a second step in here. So we have to go from the positive single-stranded DNA, and we need to make the negative strand first. So now we have a negative single strand of DNA. And what enzyme does that if we're going from DNA to DNA? The enzyme is Dna Polymerase, which is our little triangle. So we went from single-stranded, positive single-stranded DNA. We made the negative single strand of DNA. That's not what we need. We need the original form. So our friend Dna Polymerase comes in again because we're going from the DNA to DNA, and now we can make that positive copy of single-stranded DNA. This is what we want because that's what we started with. We started with a single strand of positive and we ended with a single strand of positive. And now this can be packaged into this, and the new virus can go infect another host. I hope that by going through this very slowly and with all the different colors is helpful. All right, we have one more type of DNA virus. We have our negative single-stranded DNA virus. So this time, we start with a negative single strand of DNA. And we do that in the red because the red is the negative. So that's what the virus is giving us. It's going to go across and into the host. Can the ribosome read negative single-stranded RNA or DNA? No, it cannot. We need this single-stranded positive RNA. But in this case, we have the negative strand. We're going from a negative strand to a positive strand. So that's in our favor. And it's the same process as the first one. Remember the first one? We needed to make the negative into a positive. It's the same thing. It's the same process here, except we're only starting with just the one strand. So we're going from DNA to the RNA, going from DNA to the mRNA. If we're going from DNA to RNA, we are using which enzyme? The Rna polymerase, because we're making RNA. So we can make our positive single strand, otherwise known as mRNA, which is what we need. Ribosome is waiting here for it. It reads that strand and it makes the capsids and the spikes. It is exactly the same as this process, okay? Because we're taking the negative strand and we're making the positive RNA strand. Same with here. Just because we have both strands, that's fine. We only really needed that one. Okay, so these are the same process. There's no second step. Now, we want to package the original genetic material. So our original genetic material is the negative DNA strand. We need to make more negative, but can we make a copy of the negative with the negative strand? No. There's another step in there because negative strands can only make positive strands. We're going from single-stranded DNA to single-stranded DNA. So we're going from DNA to DNA. DNA to DNA is the Dna Polymerase Enzyme. It sits on the template. It's going to make a positive strand. Do we need that positive strand? We need it to make the copy, but we can't. This is not how the original form was. The original virus is a negative-stranded RNA or negative-stranded DNA virus, so we need to have DNA polymerase come in again, sit on that template, and it'll make the original negative single-stranded DNA. We started with negative-stranded single-stranded DNA. We ended with negative single-stranded DNA. We packaged that up with these capsids, and the virus is good to go. All right, that's the DNA ones. Okay. Now, so when I say big picture, I'm talking about just a couple rules. This would be really difficult to memorize step by step without understanding the overall arching rules that are being applied. And what are those rules? The rules are, 1, we need to end, we always see, we ended with positive single-stranded RNA. We ended here with positive single-stranded RNA. We ended here with positive single-stranded RNA going into the ribosome, going into the ribosome, going into the ribosome. This is the only form that can go into the ribosome. How we got there is what is the difference. And that's the second rule. The second rule is that if you have, so that's the first rule. The second rule is a positive strand template can only give you a negative strand. If you start with a negative strand, you can only get positive out of it. So if you're trying to duplicate DNA and you're trying to go from a positive strand to another positive strand, and you're only given a positive strand, you can't get there directly because a positive strand can only make a negative strand. But then that negative strand can make the positive strand. So do you see how we go from that? We start with a positive, we need to end with a positive, but there's a middleman in the middle because of the rule that positive strands can only produce negative strands and negative strands can only produce positive strands. That's the second rule. And the third thing that you need to memorize for these concepts are the different enzymes. These are the enzymes. So those are the three things you need. If you understand those three things, you can do any of this work, any of it, without, completely killing your brain. All right, let's do the RNA viruses now. I'm going to leave out the retroviruses only because they're so strange. You can watch the video about retroviruses, but we're just going to do the three types of RNA viruses. There's our membrane. Okay. So just like DNA viruses, RNA viruses can come double-stranded. And when they come double-stranded, they have this positive RNA strand and they have a negative RNA strand. Remember, we're starting with RNA. This is all about starting with RNA, not DNA. I am going to find a Post-it note. I should have had this Post-it note when I was doing the last one, but I'll just add it. Okay. This is just going to remind us of that first rule, okay? So we're starting off with two strands of RNA. The virus is giving us a positive and a negative strand. They're twisted together, and that is what the virus is giving the host cell. Okay. Now, it's going to enter the host cell. And what's going to happen is, remember, we need to make, in order for the ribosome to read it, we need to make mRNA, which is positive single-stranded RNA. So we need to make this from what the virus gives us. And lucky us, if we apply the second rule that a negative strand produces a positive strand, we can take this negative strand. This negative strand will now be our template, and it can make from that template what we need, which is the positive single-stranded RNA. Okay, so excellent. We have what we need, but we need an enzyme. We're going from RNA to RNA. And if you remember, eukaryotes don't have an enzyme that does this. So where is this enzyme coming from? It has to come from the virus. And the virus is the one that has to provide it. And that enzyme is called RNA Replicase. So when the virus is packaged up in its capsid up here before it enters the host, it's going to have the genetic material, and it's also going to have the enzyme packaged in with it. Okay? Only RNA viruses are going to have a special enzyme. So we need this enzyme because the host cell doesn't have a way to make it. So when the virus injects the DNA or the RNA, its genetic code, it also injects the enzyme. And that enzyme sits on the template and it makes the RNA. So we're going from RNA to RNA. We need that enzyme. It has to come from the virus. And then we have our strand. Okay, so that's what we needed. So here's the ribosome waiting. It can read it now. And here is something that is interesting. So it makes the capsids and it makes the spike proteins. But it also has to make that enzyme. So it's just like a bonus recipe. So like say you were making a chocolate cake that had a crust, the cake, and then the frosting, and then maybe this one you added a layer of fruit on top. It's just an extra recipe, so this is going to make more of the enzyme too. So then when the virus packages, it grabs that more of that enzyme so that the new viruses have it. Okay, so the next step is taking the original form of the genetic material, which is double-stranded RNA, and making more of that to package into the newly formed virus. We are going from RNA to RNA. We're taking RNA and we're making exactly the same thing, RNA. What enzyme do we need? RNA to RNA, we need the replicase enzyme. And remember, the virus already gave it, it injected it to us. We just pull it from here. And what's going to happen is one of them is going to sit on the negative and one of them is going to sit on the positive. The positive will act as a template for a new negative strand. The negative will act as a template for the new positive strand. And then we've got our new negative and positive. We can package up the new genetic material into here, and then the virus is complete. So when this packages up, it packages up the double-stranded RNA, and we can't forget to package the enzyme to go with it, and then the spikes. Okay. So just a couple little changes because of that whole enzyme business. What if we just start with a single strand? You'll like this one, I promise. If we start with a single strand of positive RNA. Okay, so just like our double example, except we're just starting with one strand. And look, it's green, okay? This is good. So we're starting with a positive strand of RNA. What do we need? We need a single-stranded positive RNA in order to start this whole process. Ooh, this just can come right into the host cell. Nothing has to happen. No enzyme has to come in. It's ready to go because it's in that language that the ribosome can read already. So absolutely nothing needs to happen. It can go directly to the ribosome. The ribosome can read, and it will make the capsids and the spikes. And there you go with that. So to put in here, we need more of what we started with. So we started with one single strand of positive RNA. We're going from RNA to RNA. What enzyme do we need? We need the replicase to sit on here and to make more. Where is that coming from? You would think I would say it's coming from packaged within the virus, like this one, right? That's what you think I would say, but I am not going to say that because this is a unique situation here. The the genetic gene, the code for this enzyme is actually encoded in this genetic code. Here's the gene. Since the host can read this without any kind of enzyme needed, what happens is that when it reads that gene, it makes the replicase. We don't need the replicase to start off the reaction like we had to over here. That's why it had to be packaged, because we needed it right away. We couldn't wait. This couldn't happen until this happened. And in order for this to happen, it had to come with it. But this one, this can already happen, so we're just going to make the cell do it. And once the cell does it, it can travel over here. And we use this positive strand as a template. Positive can only make positive can only make negative. So what we end up with is a negative strand of RNA. Is that what we need though? We're starting with a positive strand of RNA. This is what the virus started with, and that's what it needs to end with. And right now we're stuck with the negative strand because that's the only thing that can happen. We're still going from RNA to RNA, so we're going to do that replicase again enzyme, and then we'll use this as a template to make the positive single-stranded RNA, ending with the same thing as we started, which is what we want. This gets packaged up in here. So the only thing that gets packaged in this one is the single-stranded RNA. We don't package the enzyme because of this unique situation where the enzyme can be produced by the ribosome, because we don't need it in this step. So it's a little easy, but a little bit you have to think. All right, the last one we're going to talk about is if we were given a negative strand of RNA. So this is RNA. What do we need? We need it to be in this form. We need it to be in positive single-stranded RNA. So we're in luck. So we're starting with a negative, and we need to go to a positive. So this can just be inserted into the host, and negative makes positive. So we can go right ahead and use that as a template to make the positive single-stranded RNA that we need for the ribosome. But what enzyme do we need here? We're going from RNA to RNA. We need the replicase. And since it's the same as this first one, we have to bring in the replicase with us. So it needs to be packaged with the virus, and then it comes in at the same time as the genetic material. It sits on the strand as the template, and it makes the single-stranded. We need to bring it this time. The ribosome is waiting. We'll read the mRNA. We will get the capsid. We will get the spikes, and we need more of this to package, so it'll make more of the enzyme to package in the new virus. The last step is to make sure we have more genetic material to package into the capsids. So we need to take our negative strand. This is negative RNA, single-stranded. We need more negative single-stranded RNA. Can we go from negative to negative? No, we cannot. We need to go into that intermediate first, so we can use our negative strand as a template to make a positive strand. We're going from RNA to RNA, so our friend Rna polymerase comes in, from up here, comes down here. Do we need the positive strand to package in here? No, we need the negative strand. So one last time, RNA polymerase comes in, it sits on the template, and it makes the negative single-stranded RNA that we need. We started with the single strand, we ended with the single strand. It packages up into the capsid. Capsid has the spikes. It has our single strand of negative RNA. And don't forget, we need to give this new virus the enzyme because the host doesn't have it. And then it's free to infect another host. Okay, that is like super long, but I think with all the colors, it was better than the lecture, if you attended lecture. Unfortunately, like due to how we give lectures, I can't, if I try to draw this on the chalkboard behind me in the lecture hall, I don't think the people in the back would be able to see what I was doing. So I had to try and draw it on the screen of the lecture, which was a little difficult. So I hope this was helpful. If it still doesn't make sense to you, just go back to the 3 rules. And the 3 rules are, rule number 1. In order to go to the ribosome of the host, it needs to be in this form. Rule number 2, when we convert things, if we have a positive strand, it can only make a negative strand. If we are given a negative strand, it can only make a positive strand. So if we want to go from positive to positive, something has to happen in between. We just can't go from positive to positive. The positive makes a negative, and then the negative can make the positive. Okay, that's the second rule. And the third rule is all the enzyme names. So all of the enzymes. And what they do. Okay, all right. My migraine is gone, thankfully, and we're all set. I will post this for you.